Hi, we're group number 6, and I'm Gabby.
Margin.
I'm Cecilia.
I'm Arlene.
I'm Priscilla.
And I'm Natalie.
Okay, so the papers we'll be covering today. For the external paper, we'll be covering, it's called Adaptive Evolution of Hybrid Bacteria by Horizontal Gene Transfer. And if you just hover over this, you'll find a link to it. And then we'll be covering papers 13 and 15 from this semester.
So as you mentioned before, the external paper we chose to use was Adaptive Evolution of Hybrid Bacteria by Horizontal Gene Transfer. This paper discusses how adaptive evolution is driven by horizontal gene transfer in closely related lineages of bacteria. Researchers did a parallel evolution experiment where two subspecies of Bacillus subtilis were used because they shared a common ancestral species. As they multiplied, there was a hybridization between the two subspecies, a largely diverse genome, and a new hybrid microbe was created. This was due to horizontal gene transfer, or more specifically, orthologous recombination. Orthologous recombination is almost the same as homologous recombination when genetic information is exchanged between two similar or identical DNA strands, but instead it just needs to have the same ancestral gene, like the two subspecies of Bacillus subtilis does. This experiment helped to conclude that both epistasis as a positive selection and hybrid incompatibility as a negative selection contribute to adaptive evolution between cross lineages of microbes. It was further deduced that horizontal gene transfer contributed to creating these complex cross lineages of bacteria due to its expanding genome and acquired new functional genes. This also led to an increased fitness amongst the hybridized microbes at the end of the experiment, opposed to the isolated subspecies present at the beginning of the experiment. As mentioned earlier with epistasis and hybrid incompatibility, horizontal gene transfer is the target of positive and negative selection. Population sweeping for positive selection when the various functional genes are attributed to bacteria via horizontal gene transfer, it's then passed on to the entire population until it's been fully hybridized, and vice versa for negative selection in hybrid incompatibility. When the bacteria is not able to harmoniously hybridize after horizontal gene transfer, the population is then stabilized by the purging out of deleterious genes from that population.
Okay, so paper 13 was titled Lateral Gene Transfer and the Nature of Bacterial Innovation. Paper 13 had to do with the idea that differences between E. coli and Salmonella are a result of lateral gene transfer. Bacteria that undergo lateral gene transfer acquire genes that are antibiotic resistant, encode metabolic properties, and have virulence attributes. However, in order for lateral gene transfer to occur, there are 3 requirements that must be met. These requirements are having a DNA transfer method from donor to recipient. Then the sequences that are acquired need to be integrated into the recipient genome. And once integrated, the genes need to be expressed properly. The paper also discussed how lateral gene transfer results in the diversification of bacteria. It has an impact on the phenotype of the species, which can be positive or negative. With these changes in phenotype, there are several advantages the organism can obtain, but it can also become a problem.
So how does this relate to the external paper? First and foremost, both paper 13 and the external paper, the focus of both is to understand how horizontal gene transfer has contributed to adaptive evolution. And both papers focus on 2 model lineages. For paper 13, we mentioned it's E. coli and Salmonella enterica. And then for the external paper, it was 2 different sublineages of Bacillus subtilis. One thing that's important to note is that when calculating how much of the genome was due to horizontal gene transfer, the external paper found that about 12% of the core genome in the first 200 generations was due to horizontal gene transfer. In paper 13, they estimated that 10 to 16% of the E. coli genome was due to horizontal gene transfer. In both cases, these numbers are similar to the total unique DNA in each bacterial strain. Okay, now going to what they find in their data. Both papers find that gene transfer is the dominant mode of evolution. Paper 13, they state, when discussing transformation, transduction, and conjugation, they state, through these mechanisms, virtually any sequence, even those originating in eukaryotes or archaea, can be transferred to and between bacteria. And then in the external paper, they find that horizontal gene transfer by orthologous recombination causes sequence evolution at an average rate of 100 base pairs per generation, which is much higher than the 0.002 base pairs per generation of normal mutations. And then both papers conclude that lateral gene transfer leads to increased genetic diversity. Negative and positive selection occur, but there is a net increase in fitness regardless of that.
For our second paper, we picked Article 15, which speaks about a species concept for bacteria based on adaptive divergence. To begin, paper 15, Voss states that the current bacterial species concept groups bacteria into species based on the genomic and phenotypic similarities between strains, which does not take evolutionary theory into account, which is where Voss's new approach to the classification of bacterial strains is expanded on. To be more exact, such approach revolves around the adaptive divergence species, which is indeed influenced by the evolutionary descent, which is where a Sable ecotype model was developed by Cohan and consists of viewing a single clone that diverges into clusters of clones that was induced by neutral mutations and genetic drift. Voss is able to test the models by implementing the McDonald-Kriepman test. The test itself serves the purpose of determining if clusters are indeed of separate species by using the neutrality index calculations. To expand on the subject, clusters are classified as different species if the neutrality index is below the number 1. Not to mention that the McDonald-Kriepman test identifies 2 types of nucleotide substitutions. The first one being synonymous and the second one being non-synonymous substitutions. To continue, isolation by distance occurs and is a phenomenon which nucleotide changes becomes fixed in geographically isolated populations. This does not result in the formation of many different sequence clusters. Geographic isolation is not expected to be significantly reduced to significantly reduce combination. It should be noted that the genes that do not contribute to adaptive divergence have no fitness consequences. The adaptive divergence concept would not be applicable to eukaryotes if there would be a low percentage of protein coding sequence, as well as a less extensive sequence evolution and a smaller population size.
Paper 15 relates to the external paper because both papers focus on the sequence divergence. The paper 15 experiment compares sequence clusters and focuses on classification of different species. The author talks about the importance of the adaptive divergence concept. The MK test was used to detect whether the 2 sequence clusters are different species through the neutrality index. Moving forward, the external paper experiment maps genomes wide horizontal transfers between 2 bacillus lineages of 7% sequence divergence. The author talks about the importance of horizontal gene transfer and how it plays a big role in bacterial evolution. LGT accelerates adaptations of bacteria to different ecological niches and import and integration of DNA from its environment.
What are the conclusions we can make via the connections between these articles? Well, we see how lateral gene transfer influences bacterial evolution, adaptive evolution, and has contributed to bacteria diversification. We see how it contributes to bacteria evolution by purging deleterious genes by contributing to the increased fitness in hybrid bacteria. And LGT also allows for bacteria to acquire genes that are beneficial for its survival, such as antibiotic resistant genes, genes that are involved in metabolic properties, and genes that have virulence attributes. We also see how LGT is a mode that accelerates generation of hybrid bacteria. And as well, lateral gene transfer influences not only bacterial evolution, but also adaptive evolution by speeding up bacteria adaptation to new ecological niches. And it also does this by utilizing transformation by importing and integrating DNA from its environment through homologous recombination. We also see how lateral gene transfer is a key factor in bacterial diversification by how bacteria can acquire new phenotypes, and also it is done by the involvement of point mutations modifying bacterial genomes, which gives them each unique differences.